Okay, so did you have any doubts in the previous topics that we had
No,
discussed?
no, ma'am.
Very good. All right. Now talking about megasporogenesis, just like how microsporogenesis was there, that was for the formation of microspores. Same way, megasporogenesis will be for the formation of megaspore. Now here what happens? The megaspore mother cell, that is MMC, that undergoes meiosis to form the gamete. Okay, now the megaspore mother cell is differentiated by the ovule in the micropylar region, meaning in the region of the micropyle, the ovule gets differentiated. Okay, now after that, what will happen is that this megaspore mother cell, it is a large cell containing dense cytoplasm and a prominent nucleus, meaning it has a thick cytoplasm and it has a prominent nucleus, means it has a very showy nucleus. Showy means that you can like easily, like you can say, visualize the nucleus. Okay, that is a prominent nucleus. Now, this megaspore mother cell, it undergoes the meiotic division to form four haploid megaspores. Okay, like we discussed in the previous class, that out of the four, only one is functional and the other three would degenerate. Now, the functional megaspore would develop into the female gametophyte, that is the embryo sac. In the case of female plants, the female gametophyte is the embryo sac. Now, since only one of the megaspore is developing the whole entire embryo sac, so we'll call it as monosporic development, right? Now, see, in the picture, we have seen how the embryo sac has a certain arrangement. Okay, so there is like a lot that goes behind it to arrange it. Let us see how the arrangement happens. See, first of all, the formation of the embryo sac. Let us see how it gets formed. Now, the nucleus of the functional megaspore, that one megaspore that was functional, its nucleus, after, like you know that these megaspores, they are formed after the meiosis has been done, right? Now, meiosis does not need to happen, isn't it? So now mitosis will take place. Mitosis is a normal cell division which increases the number of nucleus and increases the number of cells. Meiosis decreases the number of chromosomes, but mitosis does not like affect the chromosome, but it increases the cell. Clear? Like you might have studied in class 11, mitosis and meiosis, remember?
Yes, ma'am.
That concept is clear
Yes, ma'am.
or forgotten?
Forgotten a bit, but now I remember it.
Yeah. So just like I like the whole entire thing, if you just, if you want to dive deep into it, so just try to go and look back at mitosis and meiosis and how it's different. Okay, then you'll understand it if you are having problem. Now the nucleus of the functional megaspore, now it will divide mitotically because now it doesn't need to form any more haploid cells. So then it forms two nuclei. One after one division, two nuclei are. Do you know the difference between nuclei and nucleus?
No.
Okay, wait, I'll tell you more. Do you know the differences in between them?
No, ma'am.
See, bacteria is a plural word. Bacterium is a singular. Okay, when you're talking about one bacteria. Okay, and nuclei means multiple nucleus. And
Okay.
nucleus means only one. It's
Okay.
plural and this is singular. Okay, that's just the difference. So when I say that two nuclei are formed, now you can already see there are two, right? And what they do is that they move opposite to the four poles, forming two nucleate embryo sac. So it's sort of the situation, for example, this is a megaspore and it has a nucleus. Okay. Now it will divide and it will form two nuclei, like I told you, one, two. Now what will happen is that these nuclei will move to the opposite pole, sort of like this. One will go here, one will go here. Got it? Now let us see. Then after that, two more divisions or mitotic divisions take place. So from 2, the nuclei becomes 4, and from 4, the nuclei becomes 8. So ultimately, how many nuclei do we have? 8 nuclei, but there's one cell. Now, if you think about it, one cell, 1, 2, 3, 4, 5, 6, 7, and 8. One cell and 8 nuclei. Do you think this is a normal condition?
No.
No, you're right. So what should be like, what is the ideal number of nucleus in the cell?
4 or 2.
Ideal number, ideal, like normal case.
1.
Yep, 1. So this is not a normal condition. Okay.
Okay.
Now, this time, now, do you see that constantly from then and up until now, we have discussed that nucleus is dividing. Have I ever said that the cell is dividing?
No.
No. So you understand that whenever a nucleus will divide, the cell also has to divide so that both the new cells will have each of their own nucleus. Okay.
Yes.
So here, this type of mitotic division is known as free nuclear division. Why? Because the nuclear division is not followed by the cell wall formation. Meaning the nucleus is dividing, but the cell is not dividing.
Okay.
Okay. Now,
Yes.
for example, if there is this one cell with these many nuclei in it, now let's just say a cell wall is formed like this. So now this becomes cell number 1 and this whole entire thing is cell number 2. Always remember that cell wall formation, it demarcates or separates the whole entire content. Got it?
Yes.
Now, the cell walls are formed after the 8 nucleate stage, meaning once the cell has the 8 nuclei present in it, finally, like after the whole entire mitotic division is done, then the cell wall formation will begin. Okay, so that
Okay.
arrangement can happen. Now, see this. This will explain, like, clear all of your doubts. See here. Now, when we start, wait, let me just clean it. I've taught this to other batches also, so the diagram is dirty. See this now. First of all, before we move here, do you know the difference between haploid and diploid?
Haploid means
Haploid means?
it is divided from one cell, and diploid means 2 cell.
No. See, whenever we say haploid or if we say diploid, it is with respect to the number of chromosomes.
Okay.
Okay. So, number of sets of chromosomes. Do you know how many chromosomes we have, human beings? 23?
23.
Only 23. 23 pairs.
23 pairs.
So we have 46 chromosome in total. So if I say how many pairs we have?
23.
Yeah, 23 pairs, right? So two sets, like one set of 23 and another set of 23. So what are we? Haploid or diploid?
Diploid.
Very good. We have diploid. Why? Because we have two sets of chromosomes. Okay.
Yes.
Now there are some organisms like our gametes. What is the gametes of human beings?
Haploid.
Yeah, what are they? What is the name of the gametes of human beings?
I don't know.
You know this. Gametes that fuse and form a zygote, then that zygote becomes a baby. Not ovary, ova,
Ova.
and sperm. Right? Male gamete is the sperm, female
Yes.
gamete is the ova. So always remember that in the case of diploid organisms, the gametes will be haploid.
Okay.
Meaning If there is an egg, that will be N, meaning only one set of chromosome will be there.
Okay.
It won't have two pairs. It will have one pair. So this is the only difference. Now you understand? Diploid
Yes.
means two sets of chromosomes. Any two set. Like we have 23 pairs. Other organisms can have 16 pairs, 8 pairs. So that varies from organism to organism. But if any organism is diploid, then what happens? They have two sets of chromosomes. And if the organism is haploid, they will have one set
One.
of chromosomes. And in general, no matter if it is plant, no matter if it is animals or human beings, all of the gametes will always be haploid.
Okay.
Okay.
Yes.
Now, can you tell me why? Think and tell me.
Uh, because there is female and male gamete, and it will make two pairs
Yeah.
eventually.
Yes, very good. Exactly. For example, if N that is a sperm and N that is the egg, when they'll fuse, it will become 2N, isn't it? Originally, the parent was 2N and now the offspring will also be 2N. Now, had the gametes not been haploid. So imagine 2N sperm fusing with 2N ova. That will become 4N. But the parent was 2N. Getting my point? This is why the gametes are always haploid. Why? Because they have to restore the diploid number once they fuse. Clear?
Yes.
All right. So the same exact thing is for plants also. But they have a lot of, you can say, different variations too. Now let us see. Now this is the whole development of megaspore. And what is a megaspore? Megaspore is a,
Megaspore is the uh,
or not megaspore. Tell me what is embryo sac.
embryo sac is made from a single megaspore. When
Correct. Correct.
yes,
Yes. What do you call the embryo sac? Like for example, if someone could come around and ask you what is an embryo sac, so your answer should be it is the female gametophyte of plant. Okay. All right. Now see, you told me that from one megaspore only this whole entire thing develops. Correct. So this is that one megaspore. Now it will undergo meiosis, meaning what will be the ploidy of megaspore? Will it be, and see this haploid, diploid, this is ploidy. So
okay,
if I ask what will be the ploidy, so you are supposed to say haploid or diploid or whatever it is. So what will be the ploidy of megaspore?
haploid,
Megaspore is undergoing meiosis. So what will be the ploidy of megaspore?
haploid.
No. See, one more thing I'll tell you. All the cells that undergo meiosis, they will be diploid. Then
Okay,
only they'll be cut into halves and made haploid, haploid, haploid.
okay,
If already haploid is there, then what will be half? Getting my
okay.
point? So, and it's
Yes,
written here also that the megaspore mother cell is diploid, meaning it is 2N. Now,
okay.
this megaspore mother cell, you can say it undergoes the process of meiosis, and at the end of meiosis, 4 haploid megaspores are formed, right?
Yes,
Now, out of these 4, the 3 is going to
yes,
degenerate. Only one will be functional. So now this one functional megaspore, you already know that it is the haploid condition. Now do you think will it undergo meiosis?
yes.
Why?
To form the cells, nucleus.
But my question is, will it undergo meiosis? Right now I told you, right? Anything that is
Not
haploid, yeah. So it
go.
will undergo mitosis.
To,
Yeah. So now this megaspore, this will undergo the meiosis 1. Like I told you, it will form 2 nuclei, but the cell will be 1.
yes,
Then it will undergo second mitosis and form 4 nuclei, but the cell will be 1. And
yes.
then it will undergo third mitosis. And how many nuclei will be formed? 8 nuclei, but cell will be 1, right? Now
Yes.
what will happen? Those 8 nuclei that were randomly present, now they'll start to get arranged. They'll start arranging themselves. Okay. And
Yes.
ultimately, after the arrangement is done, do you see how these boundaries are there? Do you see?
Yes.
This
Yes.
doesn't have the boundary, but these have, isn't it? So
Yes.
this tells us that the cell wall has been laid out. So if the nucleus is surrounded by a cell wall, then will you call it a cell or will you call it a nucleus?
Cell.
Very good. You'll call it a cell. This thing is clear? The
Yes.
development?
Yes.
Any doubts anywhere?
No.
Any doubts in haploid diploid?
No.
Okay. Now, out of these 8 nuclei, 6 are surrounded by the cell wall and organized into cell. Like how you told me that they will be called as cell. Now, the remaining 2, do you see that in the middle they did not get any cell wall? Right?
Yes.
They will be called as the polar nuclei. They have no cell wall. Okay? Now see, this is the array. Now many a times this can be asked to make in the diagram. Like draw the structure of a mature embryo sac. So you'll have to draw this. Okay? So now let us see how the distribution is happening. Okay, let us see how the cells are getting distributed and what is the function of each of the cells. Do you see 3 of the cells? 1, 2, 3. 3 of them are here and we call them as antipodal cells. Okay.
Okay.
Below these 2 cells that you see, we call them as the synergids.
Okay.
And then, do you see one cell hiding behind?
Yes.
That is the egg cell. This is where the male gamete will come and fuse.
Okay.
This is the egg cell. And in the middle, they did not get any cell wall. So these are the 2 polar nuclei.
Okay.
Now count the number of nuclei and tell me how many nuclei are there? Nuclei.
6. No, 2.
Nuclei.
2.
Nuclei.
2.
See again. Number of nuclei I am asking. I am not saying polar nuclei. I am saying nuclei.
6.
How? Tell me how.
Each of the.
Each of the ones that have the cell wall.
Yeah.
Listen to my question again. Tell me the number of nuclei, meaning the nucleus. How many nucleus can you see?
1.
No, dear. See, all of the red ones are the nuclei.
Yes.
So how many nuclei are there?
8.
Yeah, 8 nuclei are there. Okay.
Okay.
I wasn't asking about the cell. I was
Okay.
asking about the total number of nuclei. So always remember 8 nuclei are there. But how many cells are there now? You tell me.
6.
See it again. Pay attention to all the lines.
5. No, 6.
6.
Yeah.
Again, pay attention to all the lines. See the whole diagram.
1, 2, 3, 4, 5, 6. 6 cells.
This you are saying one cell. This
Yes.
you are saying another cell. 2,
Yes.
3. Right?
3.
This
Yes.
one is 4, 5,
5.
6. What about
6.
this one?
Okay, this is also a cell.
Yeah, that is why I told you. See the whole entire diagram. See all the lines. So how many cells?
7.
How many nuclei?
Nuclei are 8.
8. So this is like a very unique property and this is one of the most common question that what is the nature of mature embryo sac? So you will say that it is 7-celled, 8 nucleated.
Okay.
They have 7 cells because don't you think it is bizarre that if 8 nuclei are there, then 8 cells should be there, isn't
Yeah.
it? But it
Yeah.
has 8 nuclei, but only 7 cells. Clear? This is clear?
Yes, this is clear.
Okay. Now let us see how the distribution is like. Now three cells are present at the micropilar end. See, the lower end is the micropilar end. Okay. And opposite to micropyle is which end?
Chazala and
Chalazal end. Very good.
Chal-
And out of micropyle and chalazal, which one would you call as the top and which one is the basal end?
Chalaza is the end basal part, and
Very hmm,
microfile is the opening.
top part. Yes,
Top.
very good. You've been studying.
Yes.
Very good. Proud of you. Very nice. Okay. Now see, the three cells that are present at the micropyle end, we can think that this is the lower part, isn't it? But it's not, right? This is the top part, the bottom one. So three cells are present, out of which two of them are the synergids and one egg cell is present, right?
Yes.
Now, do you see in the diagram that the synergids, they have certain lettuce leaf looking like structures?
Yes.
Those are known as filiform apparatus. Okay. Now they
Okay.
have a very important function is that whenever pollen tube will come, they will guide the pollen. Okay.
Okay.
It's like it is helpful in guiding the pollen tube. So this is there. And then the three cells that are present at the chalazal end, that is at the basal end, we call them as antipodal cells. And
Okay.
in the large, and you know, this one, this the seventh cell that I told you about, this is known as the central cell.
Okay.
Okay, it is the central cell, and in the central cell there are two polar nuclei.
Yes.
So the most important part, a typical angiosperm embryo sac at maturity is 8 nucleated and 7 cell. This is its characteristic feature. Got it?
Yes.
Any doubts in the arrangement?
No.
So this is how the arrangement is like. And now the thing I was telling you about, like I told you, tell me what are the three parts of the pistil?
Stigma,
Hmm,
ovary.
very good. So this part is the stigma
Yes.
and this part is the style and the basal bulgy part is the O. Now see, what happens, like I told you, the dehiscence of anther takes place. So the pollen grains, they like sort of are released into the atmosphere. Now these pollen grains, they will come and land on the landing platform that is the stigma. Now
Yes.
once it lands and inside the ovary, ovule is present and then the embryo sac is present, isn't it? Just like this. And this is the filiform apparatus. Now what will happen that this pollen grain, it will develop a tube. Okay, it will germinate. Okay, it will develop a tube and this tube will be carried like this. It will come to the micropyle area like this. And then it will release the male gamete with the help of the filiform apparatus. This is how the fertilization event is happening.
Okay.
Okay, that is why we say that the filiform apparatus in the synergid, they are responsible for guiding the pollen tube inside. Got it?
Yes.
All right. Now you can note it down. What is the last thing you've written?
Megasporogenesis.
This part is completed.
Yes, that part is completed.
Okay. Formation of embryos. Okay, you can switch off your video. Write down comfortably.
Done, ma'am. Done, ma'am.
Done, ma'am. Done, ma'am.
Done, ma'am. Done, ma'am.
Okay, any doubts up until now?
No, ma'am.
All right. So now after this, we have seen how the gametes are made, right? We have seen how the gametes are organized, the cells are formed, and all of these things, right? Now what we are going to see is that after the gametes have been made, now we are going to see how the gametes get transferred. Okay? Because if the gametes do not meet each other, then how will they fuse? Right? So now we are going to see how the gametes are going to get transferred. Now, for seeing how the gamete gets transferred, there is a proper process, which we call as pollination. Now, talking about pollination, so it is a process in which the pollen grains from the anther, they get transferred to the stigma of the pistil. Okay, like starting from the anther, they get transferred onto the stigma, which is the landing portion of the pistil, and we call this whole entire process as pollination. Now, the pollination, they can be of three basic types, which depends on from where the pollen grain is coming. Okay, there are three types of pollination: autogamy, geitonogamy, and xenogamy. Now, auto means self, meaning in this case, what will happen? The pollen grain will be from the same flower. For example, if this was a flower, from here only the pollen grain came and onto the stigma. So this will be the self-pollination. Okay. So the pollen and the egg, they will be genetically similar, isn't it? Because since they are from one flower. So the transfer of pollen grain from the anther to the stigma of the same flower, mostly in the case of bisexual flowers, it is observed within the same flower. Now see, I'll tell you that there are two types of flower: unisexual and bisexual. Now, unisexual, like I told you, it would have either male part
Or female part.
or female part. But bisexual means it will have both in one flower. Here, male flower will be different, female flower will be different. Here, one flower consisting of male part and female part. So, talking about here, so mostly if you think about it, in the case of bisexual flower, autogamy will naturally take place, isn't it? Now, there are some plants such as viola, which is commonly known as common pansy, oxalis, and commelina. These three plants, they produce two types of flowers: chasmogamous flower and cleistogamous flower. Now, have you ever seen a tulip?
Yes.
Then it's mostly like this, isn't it?
Yes.
After you open it, then it becomes like that structure. But mostly it is like this. It is a closed flower. Now, have you seen a sunflower?
Yes, yes. It
How
is
it
an
is like that, isn't it? It's open. So
yes.
the flowers can be open or they can be closed. Now there can be many examples. Now those flowers that are open flowers, meaning they are exposed, their anther, their stigma is exposed to the environment, we call them as chasmogamous flowers. Chasmogamous, okay? And all those flowers that are closed, meaning if the flower is closed, then the anther will be inside, stigma will be inside, isn't it? So what do you think? Which kind of pollination will take place in them? The closed flower. Autogamy or self-pollination. Very good. Now see, all those flowers that are closed, meaning their anthers and stigma, they are not exposed and the flower is closed. So we call them as cleistogamous. Okay. All right. Now you note down till here.
Done, ma'am. Done, ma'am.
Okay, any doubts?
No, ma'am.
All right. Okay, so we'll keep it till here only. And
Okay.
one thing, the way you are studying,
Yeah.
the way you